I know they say nothing is ever free, but this is literally free. Now, let me explain. So, as a competition for work, I can receive $1 for every lead, which is an email and a name, that I generate, and that's up to $500. Now, this money is going to be given to me in the form of an Amazon gift card. However, I know what it's like to be a broke college student trying to study for the MCAT, so I want to pass this money on to one of you guys. So, what I'm going to do is I'm going to raffle off the Amazon gift card I received from the competition. This could be up to $500 if at least 500 people register for the giveaway. But if 10 people register, that's going to be $10. If 20 people register, that's going to be $20, and so on and so forth. So if you want to sign up for this giveaway, go to the show notes for this podcast, follow the link, enter your first, last name, and email, and you'll be entered. It's that easy. All right, now to the show.
Welcome to the MCAT Basics Podcast, brought to you by the physicians at Med School Coach. Each week, Sam Smith breaks down high-yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host, Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast covers DNA mutations and DNA repair. I'll first talk about what causes DNA mutations and what types of mutations can occur. Then I'll get into how these mutations are repaired. And I'll talk about how mutations can either be repaired during replication or after replication. And this material is going to show up in 1 out of the 4 sections. This is the biochem section. With that said, I'm going to jump into it, and I hope this podcast is helpful in your studies. Also, I should say thanks to the listener, Melinda, for recommending this topic. All right, so the first thing I want to cover here is just the general concept of what a mutation is. So a mutation is an error in the DNA sequence encoding a protein. So for example, let's say that part of the sequence of a small protein is AAGGTT. If part of the sequence were to become mutated, that could mean that it was just really damaged, or it could mean that the actual genetic sequence was changed. Maybe now it becomes AAGGAA. That's pretty simple, and I'm sure you have a relatively good understanding of what a mutation is. But what causes these mutations? Well, there are many different things that can cause DNA mutations. And I'm going to break these down into 2 categories. Number 1, mutations that occur during DNA replication. And number 2, mutations that occur before or after DNA replication. Basically, mutations that don't occur during DNA replication. All right, let's start with mutations that occur during replication. Mutations that occur during replication are typically caused by DNA polymerase. Remember, DNA polymerase is the enzyme that helps replicate our DNA. It uses a template strand to synthesize a new complementary strand of DNA. However, as you may or may not know, DNA polymerase, unlike me, it's not perfect. Approximately 1 in every 100,000 bases DNA polymerase adds is the wrong base. So every time DNA polymerase adds in the wrong base, it causes a mutation at that spot where the incorrect base is added. Another way that DNA polymerase tends to screw up during replication is when it slips. So there's this phenomena called slipped strand mispairing or replication slippage that happens when DNA polymerase, as the name suggests, slips off the DNA strand and then replicates a little bit more than it's supposed to. Here's how this happens. First of all, you should know that this only happens at repeated sequences within the DNA. So pretend that the polymerase has just synthesized the repeated region ATTCG, ATTCG, ATTCG. So that's the same sequence repeated 3 times, like stuck right next to each other on a strand of DNA. Now, the first thing that happens here, as I said, is that the DNA polymerase falls off, or it kind of slips off the template strand. The next part is a little bit hard to understand, but basically what happens is that this newly synthesized strand with the repeated region forms a loop. Why does this loop form? Well, as you can imagine, on the complementary strand, there's also a repeat that is complementary to the one that is on the strand that was just synthesized. So, what happens basically is this loop forms because the outermost repeat, that ATTCG sequence, kind of shifts a little bit upstream on the complementary strand and base pairs with that part, creating this loop that contains the two other repeats, the AATCG, AATCG. If that's making you say, huh? Ruby? There's an absolutely fantastic YouTube video titled DNA Polymerase Slippage Explained with Velcro that can really help you see this point if you're not getting it. Anyways, the next step here is that DNA polymerase comes back and it rebinds to the template strand and begins producing the daughter or complementary strand again. And meanwhile, this loop that's formed just remains still. It kind of remains as a loop for the rest of the time that replication is occurring. Thus, whatever material is in that loop is essentially going to be duplicated in the daughter or complementary strand. So this slip strand mispairing or replication slippage is going to result then in what's called an insertion. More on that in a little bit. So that's obviously somewhat of a complex topic, so let me just kind of summarize that in the most simple way. So in short, replication slippage or slipped strand mispairing happens at repeated regions in our DNA and causes duplication of those repeats. Okay, so that's kind of the two ways that DNA mutations can occur during replication. I should also mention that these mutations are pretty much all fixed, and I'll talk about that as well a little bit later on. Now, the next thing I want to get into here is mutations that occur before or after replication. In other words, I want to talk about mutations that don't occur during replication. So these type of mutations can be caused by what are called mutagens. Mutagens are agents that cause DNA mutations, and these can cause all different types of mutations. They can cause insertions or deletions, they can cause DNA breaks, they can cause point mutations, etc. They can really cause any type of mutation. Now, what I want to do real quick is just delineate between a mutagen and a carcinogen. They're two pretty similar terms, and it's something that the MCAT wants you to be able to do, is to delineate between those two and know the differences. So as I said, mutagens are agents that cause DNA mutations. On the other hand, carcinogens are agents that cause cancer. With that said, some mutagens are carcinogens, and then some carcinogens are mutagens, but there isn't perfect overlap. I'm going to give you an example here to really kind of make this clear. So UV radiation in sunlight is a known mutagen. It can mutate your DNA. So when you stand outside for like 10 minutes in the sun, it's likely that at least one base in your whole genome is mutated or damaged in some way. More specifically, UV light causes a type of mutation called a Pyrimidine Dimer. Basically, this is when two pyrimidines that are next to each other, either two C's or two T's, get hit with a UV ray and form a bond to each other. And this causes the DNA double helix to bend a little bit, and it's not good for your DNA. Now, with that said, will this single mutation lead to cancer? Probably not, right? It'll probably get repaired and everything will be okay. So in this case, when you're outside for like 10 minutes, this short-term UV exposure isn't necessarily carcinogenic. Now, on the flip side, let's say that you use a tanning bed for like 2 hours straight every single day for a whole year. So every single day for that year, your DNA is going to be mutated, and it's going to be mutated quite a bit. Will this lead to skin cancer? Maybe. I mean, it's probably kind of hard to say, but what I would say is that you're much more likely to develop skin cancer in the case where you're laying in that tanning bed every single day. So in the second case of the long-term UV light exposure, I would classify UV light as more of a carcinogen. So in this case, you have long-term UV light exposure being both a mutagen and a carcinogen. And yes, this is not the best example because UV light is a known carcinogen, but just for the sake of the argument, pretend that UV light can either be a carcinogen at high concentrations or at low concentrations. It is not a carcinogen. And the point I'm trying to get across here is that a compound can in fact mutate DNA and not necessarily cause cancer or it can mutate DNA, and it can cause cancer. So that's how a mutagen can be a carcinogen or not be a carcinogen. What about the flip side? Is there any carcinogens that are not mutagens? In other words, are there any compounds out there that exist that cause cancer without mutating DNA? The answer is yes. According to a paper by Mohn and Associates, there are a few animal and human carcinogens that appear to possess no mutagenic activity. In other words, they don't mutate DNA. These are things like saccharin, benzene, carbon tetrachloride, and cadmium. I'm not going to get into mechanisms here, for how those compounds cause cancer. I'll save that for med school. But essentially, these compounds cause cancer without mutating DNA. All right, so let me just summarize this section here by saying a mutagen is a compound that mutates DNA, and a carcinogen is a compound that causes cancer. There's a lot of overlap between these two, but they don't perfectly overlap. Make sure you know the difference between these two for the MCAT. Hopefully, this gave you a pretty good look at what the differences and similarities between them are. With that said, there are really three different categories of mutagens. This includes radiation, DNA reactive chemicals, and metals. In terms of radiation, things like UV light, X-rays, gamma rays, and alpha particles can cause mutations to occur in DNA. In terms of DNA reactive chemicals, by the way, which are just compounds that readily react with DNA, that's things like benzene, reactive oxygen species, polycyclic aromatic compounds, ethidium bromide. The list goes on and on and on. And then finally, the third category of mutagens are metals. This includes things like arsenic, cadmium, chromium, and nickel. So since all of these different compounds, the radiation, the reactive chemicals, and the metals I talked about are mutagens, they have the potential to mutate DNA. Now, what I want to do next is kind of go through some of the different ways that these actually mutate DNA. Talk a little bit mechanistically about how this works. So first off, I just want to say that all of these different chemicals and metals and radiation can really act in a bunch of different ways to cause mutations within DNA. I just want to point out a few in particular. So number one, they can actually change the identity of a single base. There are a few different ways that this can occur, including oxidation, deamination, and alkylation. For example, the oxidation of guanine leads to what's called 8-hydroxyguanine. And when any polymerase encounters this 8-hydroxyguanine, whether that's RNA polymerase for transcription or DNA polymerase for replication, it can't recognize that base. It's like, what the fuck is this? I don't know what to do with this. And that's obviously bad because then that stops transcription and stops replication, which then can lead to bad things down the road, whether that's not being able to produce a particular protein or errors in replication. Another thing that mutagens can do is create DNA adducts. DNA adducts are a piece of DNA that's covalently bonded to a chemical. So for example, acealdehyde, safrole, as well as a compound called benzoprenyl diol epoxide can actually come in and covalently bond to DNA and then cause problems in, again, transcription as well as replication. That's really no bueno. These mutagens can also break strands of DNA. They can cause bases to cross-link or form bonds with each other. An example I already talked about is when UV light causes pyrimidine dimers to form. And then finally, mutagens can cause what's called Interstrand Crosslinks. This is when two bases from complementary strands are covalently linked together due to damage to the DNA. So this is similar to like pyrimidine dimers, except for that it's like across the strands of DNA. It's from one strand of DNA to the other. And there's a bunch of different agents that can do this, including platinum compounds, nitrogen mustards, as well as alkylating agents. And you could imagine how bad these would be for both replication and transcription, right? When a polymerase comes upon a piece of DNA that's basically glued together, it's not going to be able to open up those two strands and get in there and do its job. So again, these can cause stoppages in replication and transcription. Okay, so those are some examples of how mutagens can mutate and damage DNA. Now, the next thing I want to talk about here is why these mutations matter. What are some of the things that they do? I've talked about how they cause cancer, but there's some other things that mutations can do and other ways that they can affect people's lives. There's one in particular that I really want to talk about called inborn errors of metabolism. That's a phrase that you should definitely be familiar with for the MCAT.
At Med School Coach, we know that studying for the MCAT exam can be challenging, especially for busy students on the go. That's why our team created MCAT Prep, the only all-encompassing study app built specifically for the MCAT. MCAT Prep by Med School Coach provides student access to extremely high-quality content and a personalized curriculum for free. The app has more than 250 videos, 1,000 flashcards, and 1,000 unique MCAT questions. Plus, MCAT Prep by Med School Coach allows students to create a personalized study schedule and track progress over time. You need every competitive advantage you can get to get into medical school, and now you can put the experts from Med School Coach into your pocket. It's the closest pre-med students can come to a personal tutor without spending a penny. Download MCAT Prep by Med School Coach for free at medschoolcoach.com/MCAT. Or download it directly from the Apple Store or Google Play Store. You can achieve your medical school dreams, and MCAT Prep by Med School Coach can help.
All right, we're back. And as I said before, I want to talk about another way that mutations can really affect people's lives. And specifically, I want to talk about inborn errors of metabolism. So, inborn errors of metabolism occur when someone receives a mutated gene from one of their parents. And in the context of inborn errors of metabolism, this error or this mutation is in a gene that encodes a protein that is involved in metabolism. This mutated gene then leads to some defective protein, typically an enzyme, within a metabolic pathway. Thus, whatever metabolic pathway this defective protein is involved in doesn't work properly. Now, an example of a disease that comes from an inborn error of metabolism that I came across is called maple syrup urine disease, or MSUD. This is a rare genetic disorder characterized by the mutation of a gene encoding the enzyme complex called the branched chain alpha keto acid dehydrogenase. Now, that's a complex name for an enzyme that does something pretty simple. It helps break down the 3 branched chain amino acids, which are leucine, isoleucine, and valine. Now, when this enzyme is not functional, like in the case of maple syrup urine disease, these amino acids can actually build up to the point where they become toxic. Now, I know one thing that probably is maybe on your mind, it was on my mind. Why the name? Why is this called maple syrup urine disease? Obviously, this is just for your own interest, but apparently one of the symptoms of the disease is that urine, sweat, and or earwax that come from a patient with this disease tend to smell like maple syrup. Interesting, sad, and unfortunate for patients who have this disease. Now, the important thing to know here and kind of the takeaway message is that an inborn error of metabolism is a category of disease that results from an inherited mutation in the sequence of a protein, typically an enzyme, that's involved in the breakdown of nutrients. It's involved in a metabolic pathway. So beyond cancer, that is another way that mutations can really affect the life of an organism and the life of a human. So I've talked about the general concept of a mutation, where they occur, how they occur, and what they can do. The next thing I want to talk about here is how they can be categorized. So there's a few different ways that mutations can be categorized. They can be categorized by the effects they have on an organism, advantageous, deleterious, or neutral. They can be categorized by where they occur, random mutations, translation errors, transcription errors. Or they can be categorized by the effect that they have on a protein, missense, nonsense, and silent. I want to go through each of these, introduce some terms, and just kind of explain what everything means. I'll start with categorizing mutations based upon their effects on an organism. So really, when we're talking about the effects they have on an organism, mutations can either be advantageous, meaning good, deleterious, meaning bad, or neutral, meaning neither bad nor good. Now, what does it mean for a mutation to be good, bad, or neutral? Well, really, we're talking about reproductive fitness. We're talking about what a mutation does to affect the reproductive success that an organism has. So, advantageous mutations are defined as mutations that increase the fitness of an organism. Basically, these are mutations that increase the reproductive success of an organism. Maybe that's increasing the longevity of an organism, or in some way increasing just something. They're increasing something about the organism that makes them better at reproducing. So, a good example of this is a mutation that causes innate resistance to the HIV virus. So, some people are born with mutations in the CCR5 gene. CCR5, if you're not familiar with it, is a receptor on the surface of a cell, and it's actually the co-receptor for HIV. So, HIV uses it to actually gain entry into the cell. So, when people have a mutation in the gene that encodes that protein, that CCR5 protein, HIV can't actually enter their cells. And so these people are, for the most part, resistant to HIV infection, which can help people live longer, thus reproduce more, and therefore it's an advantageous mutation. Deleterious mutations, on the other hand, are mutations that decrease the reproductive fitness of an organism. This is just a harmful mutation. The inborn errors of metabolism that I talked about earlier are a good example of this. Right? They're a group of mutations that impair a person's ability to break down or metabolize certain nutrients, leading to, in many cases, a shorter life and a decreased ability to reproduce. Finally, neutral mutations are mutations with no effect on the sexual fitness or the reproductive fitness of an organism. So, for example, say that a mutation in my genome causes 1 in 10 of my hairs to be gray. And, it's that way from birth. So all throughout my life, I've had 1 in 10 hairs gray. So all that mutation would do would give me speckled gray hair. It wouldn't necessarily impact how successful I was at reproducing, nor would it really impact my life in any real way. Thus, it would be a neutral mutation. This example was sponsored by Touch of Gray by Just for Men. No mix, no mess. Just comb in, and in 5 easy minutes, you'll get the perfect salt and pepper look. All right, let's now talk about categorizing mutations based upon where they occur. So, in part of the MCAT content outline put out by the AAMC, they include a few terms that you need to know regarding mutations. And so that's how I kind of came up with this category. The terms they have include random mutations, translation errors, transcription errors, base substitutions, inversions, addition, deletion, and translocation. So maybe more accurately than saying this is the category of where mutations occur, this is the MCAT category of mutations. So I'll start with random mutations. So my understanding is that the term random mutation is just basically an umbrella term kind of for all mutations that occur outside of translation and transcription. In other words, this is most of the mutations I've already talked about. Now, there is some evidence that apparently there is that some mutations aren't necessarily random. Like there are hot spots, I guess, in the genome where mutations can occur more frequently and therefore might not be just random. However, for the sake of the MCAT, just consider that a lot of these different point mutations, as well as mutations coming from these different mutagens, are random. So, for example, UV light that we talked about earlier causes these Pyrimidine Dimers. That would be a random mutation. Some of the other mutagens, the metals as well as the chemicals, those will also cause random mutations. Next is translation errors. So translation is the process of taking mRNA and converting it or translating it to protein. And interestingly enough, there can actually be errors that occur during that process. And that's not necessarily due to a mutation per se, but it can have the same effects as a mutation, right? It can lead to the incorporation of the wrong amino acid into a protein, which can then alter the activity of the protein. Now, errors in translation can happen in a few different ways. Number one, they can occur when the tRNA is misacetylated. In other words, the wrong amino acid is tied to the wrong tRNA. Therefore, when that tRNA is used by a ribosome to convert mRNA to protein, the wrong amino acid is going to be put into that protein. The second way that errors in translation can occur is in tRNA misdecoding. Basically, the triplet codon on the tRNA doesn't match up with the triplet codon on the mRNA, but that tRNA still comes into the ribosome and kind of matches up with that codon, even though they don't match up perfectly, and it adds the amino acid to the polypeptide anyways. So again, translation errors, not necessarily due to mutations, but can cause the same thing, and then it can lead to the incorporation of the wrong amino acid in a protein, altering the activity of that protein. Next are transcription errors. So transcription is the process of taking a DNA and converting it to mRNA. I've talked a little bit about this. This is very similar to errors that occur during replication. Basically, this would be any error in which the wrong mRNA is produced from the correct genetic sequence. In other words, this is where RNA polymerase kind of messes up and puts in the wrong RNA base and produces the wrong mRNA. And apparently, the most common form of this type of error is a C to U base substitution. In other words, RNA polymerase sees a place where it should put a C and instead puts a U or a uracil. And that, of course, can end up resulting in an error in whatever protein is then produced from that mRNA. Also, shout out my alma mater, CU, let's go Buffs. So again, just to kind of summarize that, transcription errors are errors that occur when a gene is incorrectly transcribed from DNA to mRNA and eventually gets made into a protein that is the incorrect protein. Next comes base substitution. Base substitution refers to the replacement of one nucleotide in DNA or RNA with another nucleotide. I think the best example of this is like DNA polymerase putting in the wrong base in the complementary strand during replication. It should be putting an A, like an adenine, but instead puts in a thymine. That's an example of a base substitution. And these create what are called point mutations, a mutation where only a single base pair is altered. Next, we have insertions and deletions. An insertion is when an extra nucleotide or extra nucleotides are added into a strand of DNA. This is synonymous with an addition mutation. On the other hand, you have deletion mutations. That is where a single nucleotide or multiple nucleotides are deleted or taken out of a DNA strand. And these can lead to what are called frame shift mutations. This is probably another term you should know for the MCAT. Basically, this is where the reading frame of a gene is shifted in some way. For example, say that some gene is made up of 120 nucleotides. That means that it's made up of 40 different codons, right? There are 3 individual nucleotides in each codon. Each codon corresponds to an amino acid. And so this would be a protein made up of 40 amino acids. And we're saying that the reading frame here is 120 nucleotides. Obviously, you have some non-coding regions of that gene that are upstream and downstream, but let's just take the reading frame. Now let's say that because of a mutation, you take away the very first nucleotide in the sequence of that gene. Well, what's that going to do? Well, that's literally going to shift the entire gene or the way that entire gene is read, right? Because it's going to change every single codon down the line. You're now grouping codons a bit differently. And that's going to change the whole entire amino acid sequence of that protein. Probably. It's likely that it will. The same thing would happen if you inserted a nucleotide at the very beginning of that gene. You would now have 121 nucleotides, and the 3-letter codon sequence going down that gene is again going to be completely changed, resulting in a completely different amino acid sequence that makes up whatever protein that we're talking about. Now, if you're still having trouble kind of understanding exactly how it shifts what's being read in that gene, take the following example. Take the sentence, "The big bad dog had one eye." This sentence is composed of 7 3-letter words. In genetics terms, it's basically 7 different codons, each of which correspond to a particular amino acid. Now, say I insert a single letter, D, at the beginning of this sentence, and then reform the 3-letter words or the 3-letter codons. What would the sentence become? Well, it would basically become baby talk or like gibberish. Dith, abi, jibbe, didu, oga, ade, ni, Yee. That's literally what it would become. And so you can see then how a frame shift mutation would really mess up the amino acid sequence of a protein, which can lead to really bad things. For example, according to one of the papers I was reading, frame shift mutations are apparent in many severe genetic diseases, such as Tay-Sachs. All right, finally, we have made it to some of the last types of mutations that the MCAT wants you to know in this MCAT mutation category. These are chromosomal mutations. The first is called an inversion, and the second is called a translocation. So, an inversion is a chromosomal mutation where part of the chromosome breaks off, flips around, and then becomes reattached. I kind of almost like imagine this is in my head as literally taking a chromosome, ripping out the middle portion of that chromosome, flipping it around, putting it back in place, and then gluing it back in place, but upside down. That is a chromosomal inversion. And typically these aren't too bad because you still have all the genetic material you had before. Now, if you're ripping out part of a chromosome and where you rip is like in the middle of a gene, and then you flip it around and glue it back in place, that can screw something up because now you only have like half of a gene and maybe you have half of another gene glued to that. And now you're making some random mutated protein that's not effective. So there are some cases where an inversion can be bad, but it's not as bad as some of the other types of mutations. The other type of chromosomal mutation I want to mention is a translocation. This is when a chromosome swaps bits of genetic material with another chromosome. So for example, chromosome 21 gives a few genes to chromosome 3, and chromosome 3 is like, oh, thanks for those genes, 21. I'll give you a few. And then it gives some of its genes to chromosome 21. I kind of think of it like gene trading in a way. Now, with that said, there is a very specific type of translocation that's good to know. It's called a Robertsonian translocation. And this is a bit complex, but in short, this occurs when two acrocentric chromosomes get stuck together to make one large chromosome. And by the way, the acrocentric chromosomes are chromosomes 13, 14, 15, and 22. Biologically speaking, the most common Robertsonian translocation occurs between chromosomes 13 and 14. And again, in this type of translocation, basically two chromosomes merge to form a big old chromosome. The last way that mutations can be categorized is in terms of the effect that they have on a protein, and more specifically, the effects that they have to the amino acid sequence of a protein. Now, this is missense mutations, nonsense mutations, and silent mutations. Missense mutations are substitution mutations resulting in a change in an amino acid. So, for instance, let's say that a single mutation causes a glycine amino acid residue in a protein to change to a valine. Well, that would be a missense mutation. A nonsense mutation is a substitution mutation resulting in a premature stop codon. So, for example, this could occur when the fifth amino acid in a protein that's like 100 amino acids long is mutated and it goes from coding a glycine residue to coding a stop codon. That would be a nonsense mutation. And this is bad, right? Because when a ribosome comes upon a stop codon, it's going to stop producing that protein. And so in this case, you're essentially going to produce a protein that is 4 amino acid residues long, even though it's supposed to be 100 long. Finally, you have silent mutations. These are substitution mutations resulting in no change in the amino acid sequence of a protein. So for example, this would be a mutation that causes a change in the nucleotide sequence of a protein, but no change in the amino acid sequence. Now, you might be asking, how is that possible? Well, if you remember, there are multiple triple letter codons that correspond to the same amino acid. So you could in fact change the nucleotide sequence of a gene and still keep the same amino acid sequence. And if you do that, that's called a silent mutation. Now, I remember these different names by kind of thinking about how bad each of these names sound. So nonsense versus missense versus silent. Nonsense sounds really bad, And so I remember that results in a premature stop codon, which is really bad. Like that has the worst effects. Then you have missense, which to me sounds like the next worst. And I remember that that's the next worst thing that could happen, which would be there's a substitution mutation resulting in the change of an amino acid. And then finally, a silent mutation is kind of what it sounds like. It doesn't sound very bad, and it's not very bad because it doesn't result in the change of an amino acid. All right, so that is a shit ton of stuff regarding different DNA mutations. The next thing I want to talk about, and this stuff is important too, I think it's a little bit more interesting, is the repair mechanisms that exist to repair these mutations. But first, a word from Shia LaBeouf.
Do it! Just do it!
Thanks, Shyla. So what he's talking about there is the fact that you should sign up for the MCAT Basics giveaway. Again, I am going to give away an Amazon gift card. The dollar amount will be however many people sign up for the giveaway. If 10 people sign up, that's $10. If 100 people sign up, that's $100, up to $500. So if you're interested in this free money, and hopefully what it's really meant to do is help, pay for some of the expenses related to taking the MCAT, sign up using the link in the show notes. All right, now back to the show. All right, so in the next part of this podcast, I want to talk about DNA repair mechanisms. And I'm going to break this down into 3 different categories. Number 1, DNA repair that happens during replication. Number 2, the repair of DNA mutations that don't occur during replication. And then number 3, repair of DNA breaks. I'll start with that first one, the repair of mutations that occur during replication. So as I talked about a little bit earlier, DNA polymerase makes mistakes. One in about every 100,000 nucleotides added during replication is the wrong one. Now, that doesn't sound like very much, but there's about 6 billion base pairs in each diploid cell. So that amounts to about 120,000 mistakes every single time a cell divides. Now, what happens to those mistakes? If they just accumulate, that could lead to very bad things. So how do they get repaired? Well, lucky for us, the DNA polymerase actually has a proofreading function. In other words, it can proofread the DNA it's synthesizing and fix mistakes as it goes. To be a bit more detailed here, the DNA polymerase synthesizes new DNA strands in the 5' to 3' direction. So when it's replicating DNA, right, it uses the template strand and creates a complementary strand to that template strand. And it does that in the 5' to 3' direction. However, as I just mentioned, the DNA polymerase actually has proofreading activity, and that is in the 3' to 5' direction. That should make sense, right? Because if it's going to repair a mistake it made, it's got to go backwards to do that. So it goes backwards, and it goes in the 3' to 5' direction. And this activity, by the way, is called 3' to 5' exonuclease activity. Now, what this proofreading activity looks like is that the polymerase can actually back up, remove the mismatched base, and then replace it with the correct base. Pretty simple. Now, one thing I want to point out here that I think is a little bit important is that it's not necessarily the same DNA polymerase that makes the mistake that fixes it. Interestingly enough, DNA polymerase delta can actually proofread and fix errors that are made by DNA polymerase epsilon. The take-home point there really is just that the DNA polymerase that makes the mistake isn't always the polymerase that fixes the mistake. Sometimes another polymerase can come in and use that 3' to 5' exonuclease activity to fix a mismatched base. Just keep that in mind. All right, so that's how mutations that occur during replication are fixed. Next, let's talk about DNA mutations that are not repaired during replication. So these can be either mutations that escape detection by a polymerase and aren't fixed during replication, or they can be mutations that arise from all the different sources I talked about earlier. They can come from radiation, DNA reactive chemicals, and even metals. So I want to talk about 5 different repair mechanisms here. Number 1, I want to talk about direct reversal DNA repair. Number 2, I'll talk about mismatch repair. Number 3, I'll talk about base excision repair. Number 4, I'll talk about nucleotide excision repair. And then finally, the last one I'll talk about is called Interstrand Crosslink repair, or ICL. All right, I'll start with direct reversal DNA repair. So, direct reversal DNA repair is DNA repair that does not require the breaking of phosphodiester bonds. Basically, in this type of repair, enzymes come in, they fix the DNA, and then they leave. And they do all that without, breaking the DNA or breaking phosphodiester bonds within the DNA in order to fix it. For example, this repair mechanism can help fix damage caused by DNA alkylating agents. So, when DNA is alkylated, basically an alkyl group is added to the DNA. For instance, like a methyl group can be added on to one of the bases in DNA. When that happens, that's a problem that changes basically the structure of the base or changes the structure of the nucleotide and isn't able to be recognized maybe by DNA polymerase, RNA polymerase, and affects transcription and replication. So, what enzymes can do in this direct reversal DNA repair mechanism is they can come in, remove the alkyl group directly, and then boom, you're back to your original base. So, basically, in this direct reversal DNA repair mechanism, any damage that's done is directly removed to restore the original base. The next repair mechanism is called mismatch repair. You might see this abbreviated as MMR. So typically, mismatch repair fixes base mismatches that have arisen during replication, and it fixes these insertion deletion loops that are a result of replication slippage that I talked about earlier. Now, this only occurs on the newly synthesized daughter strand after replication. And to me, that's pretty impressive, right? These enzymes that are involved in this mismatch repair mechanism are able to actually differentiate between the daughter and the mother strand of DNA. That's pretty sweet. Now, how do they tell the difference between these two? I don't know, and the MCAT obviously doesn't expect you to know, but still pretty interesting. Okay, so in mismatch repair, the mismatched base or mutated region of the DNA is removed by an exonuclease, and then DNA polymerase comes in and it resynthesizes that region, putting in the correct bases now. And then this is sealed in place or glued in place by DNA ligase. Now, I should be clear here. The removal process involves more than just the mismatched nucleotide itself. It can be a few or even up to 1,000 base pairs of the newly synthesized DNA strand. So just keep that in mind. It's not just the error that's removed, it's the error as well as some of the regions around that error. And then, of course, once all of those nucleotides are removed, then the mother strand, which is still intact, right, because these enzymes are only acting on the daughter strand, can act as a template for DNA polymerase to then come in and resynthesize the correct bases or put in the correct bases in order to be complementary to that mother strand. Now, apparently, the enzyme that's involved in this process recognizes certain sequences, like, for example, GATC, and then is able to cut at that particular sequence. So, my understanding is that this enzyme finds the closest sequence it recognizes near a mutation or near a mismatched base, and then it cuts at that sequence. That's why it's kind of a random number of bases that get removed along with that mismatch, right? Because you're cutting out everything that is between those two recognized sequences where these enzymes can cut. That's probably a bit out of the scope of the MCAT, but maybe it'll help you remember that in mismatch repair, a varying number of bases are excised and then filled in, and that occurs only on the daughter strand. Next is base excision repair. Base excision repair corrects single base mutations that don't significantly distort the DNA double helix. So these are basically single base point mutations that aren't significant enough to cause these, big kinks or big deviations in the DNA double helix. So for example, this fixes things like oxidative damage, deamination, as well as alkylation. Remember, these alterations, again, oxidation, deamination, and alkylation, slightly change the identity of the base so that RNA polymerase can no longer recognize that base. DNA polymerase can't either. That leads to faulty protein production as well as errors in replication. And since these forms of damage or these types of mutagens don't really cause, big changes in the nucleotides, they're only slightly changing the bases of the nucleotides. That doesn't really distort the DNA double helix too much. And so then base excision repair is able to fix these type of mutations. Now, mechanistically speaking, here's what happens. So an enzyme comes in and removes the damaged base from the DNA by cleaving the bond between the base and the deoxyribose sugar. And I would just want to make it clear here, it's not removing the whole nucleotide at this point. This enzyme is just cutting off that base, whether that is adenine, cytosine, guanine, or thymine. This creates what's called an abasic site. This is a site in the genome, in this case, just one specific site that doesn't have a base. Next, an enzyme that is able to actually recognize abasic sites comes in and removes the abasic site. It basically cuts the DNA backbone at that site and is able to remove that piece of the DNA backbone. So at this point, the whole nucleotide has been removed. Next, DNA polymerase comes in and fills in this gap. It uses the complementary strand as a template in order to tell which base it needs to put in. Then finally, DNA ligase comes in and seals the gap between the new nucleotide and the existing DNA strand. And voila, you have fixed a single mutated base. All right, so that is base excision repair. Next, I want to talk about something that's similar, but a little bit different, called nucleotide excision repair. So in contrast to base excision repair, nucleotide excision repair repairs bulky DNA double helix distorting damage and mutations. An example of this would be like a pyrimidine dimer. As I talked about a little bit earlier, UV light can cause two pyrimidines that are next to each other to form bonds with each other and dimerize. And as you could imagine, that kind of pulls on the DNA double helix in a way that is not natural and causes it to distort quite a bit. And so this is something that nucleotide excision repair would fix. Now, in terms of its mechanism, what happens first is that this damage is recognized. Again, this is some type of bulky helix distorting damage. After it's recognized, a 10 to 20 nucleotide long strand of DNA is removed both upstream and downstream of the damaged site by endonucleases. So if you add that up together, this is a total of about 20 to 40 bases are removed in order to fix this one, bit of damage. After that, this gap is filled in by DNA polymerase, which puts in the correct nucleotides, and then everything is sealed up by DNA ligase. So that is nucleotide excision repair. Next, I want to talk about Interstrand Crosslink repair. Now, you may have never heard of this type of repair, but the reason that I want to talk about it and the reason that I bring it up is that earlier I talked about Interstrand Crosslinks as being a form of DNA damage that you should be familiar with. Remember, this is when complementary strands of DNA covalently cross-link together. It's like gluing the two strands of DNA in this DNA double helix together. So how do you fix that? Well, you fix it through Interstrand Crosslink repair. In short, the mechanism is essentially 2 rounds of nucleotide excision repair, since again, this type of mutation affects both strands. So you do nucleotide excision repair on one side of the DNA duplex, then you flip to the other side and you do nucleotide excision repair on that side, and then you're fixed. So again, Interstrand Crosslink repair is basically 2 rounds of nucleotide excision repair. And it fixes these interstrand crosslinks, which are covalent bonds between complementary strands of DNA. Now, before I move on to the repair of DNA breaks, I want to quickly summarize all of these different repair mechanisms I've gone over so you can clearly see the differences between them. So first we had direct reversal DNA repair. This is where an enzyme comes in and directly fixes whatever damage has been done to the DNA. Next, we have a mismatch repair. This fixes mismatched bases that are a result of errors in DNA replication, and it removes kind of a variable amount of bases around a mismatch and then fills that in. Next, we have base excision repair. This corrects single base mutations that don't significantly distort the DNA double helix. And in this case, a very small part of the DNA strand is removed and then filled in. Typically, this is a single base that's removed and filled in. Then we have nucleotide excision repair. This repairs bulky DNA double helix distorting damage and mutations. And in this case, a pretty large part of the DNA strand is removed and then filled back in to fix that damage. Finally, we have Interstrand Crosslink repair. This repairs DNA strand crosslinking, which is covalent bonds between the two complementary strands of DNA. And basically, this is 2 rounds of nucleotide excision repair back to back, one on each strand. All right, so last thing here, let's talk about the repair of DNA breaks. So different mutagens can actually cause DNA to break. So these breaks can either be single strand breaks, in other words, only one of the 2 strands has a break in it, or they can cause double-stranded DNA breaks, which, by the way, are much worse and are breaks that break literally both strands of DNA. It's like taking a butcher's knife and just chopping the DNA. Now, single-stranded breaks are repaired through what's called single-stranded break repair, aptly named, and it's pretty simple, really. Enzymes come in and they ligate the DNA. They seal it right back up. And it's not only like 1 or 2 enzymes that do this. It's a team of enzymes that come in, pull the DNA back together, and then seal that break. So keep that in mind. It's a big old team of enzymes that do this, not just like a single, Superman type enzyme. Now, double-stranded DNA breaks are repaired through, again, a very aptly named mechanism called double-strand break repair. Now, as I kind of mentioned previously, breaks between both strands of DNA, these double-stranded breaks, they're very, 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 very bad. They're basically the Vladimir Putin of DNA damage. And so there are 2 mechanisms that our cells use in order to repair these double-stranded breaks. One of which is called homologous recombination, and the other of which is called non-homologous end joining. I'll start with homologous recombination repair. So homologous recombination repair is high fidelity or high accuracy repair. It uses information copied from a homologous sister chromosome that's undamaged. So basically, homologous recombination repair uses the sister chromatid of the broken chromosome as a template to repair that broken chromosome. So basically, it can go look at the site that's broken on the other homologous chromosome and say, okay, this is what it should look like, and then use that as a template for repairing that broken region. And so that should make sense then why that's pretty high fidelity or high accuracy, because you're using the homologous sister chromatid as a template for repair. So you're getting pretty accurate fix of that break. Now, on the other hand, non-homologous end joining is not as high fidelity. Basically, in this repair mechanism, the ends on each side of this break are directly glued back together, and there's no template that's used. And you could imagine then that sometimes this results in the loss of genetic material. Let's say that you have 2 breaks that occur, right? You have one break, and then a little bit downstream of that, you have another break. Well, then you're literally cutting out a piece of DNA. And if you were to then have that little piece kind of float away, somewhere else into the cell, you could then take those 2 cut ends and glue them back together, and you just lost a piece of DNA. And so as you could see, non-homologous end joining is not quite as high fidelity as homologous recombination repair. So altogether, homologous recombination repair is template repair. You're using information from a homologous chromosome, the sister chromatid, to then repair the broken piece of DNA. On the other hand, non-homologous end joining is non-template repair. The ends of the break are just directly glued back together. All right, so that is double-strand break repair. And that's also it for this podcast. I hope you found all this information helpful. Obviously, it is quite a bit of information and quite dense. So, listen to it multiple times, whatever you have to do. But all this stuff is important. And a lot of the outline for this podcast comes directly from the content outline put out by the AAMC. Also, this is the very last time I'm going to say it. But as Shania Twain said earlier, just do it. Just go enter the giveaway. Again, this is a competition for work. I'm trying to win as well as I'm trying to, provide somebody with a little bit of free money. So if you're interested, go join that. Also, as always, if you found this podcast helpful, go leave us a review on iTunes. It helps us in the rankings as well as helps us reach other students. Thanks for listening, and I'll see you next time.
At Med School Coach, we know that getting into medical school is hard. In fact, 60% of pre-meds who apply to medical school don't even get accepted. And if you want to get accepted into a top medical school, it's even harder. It's a complicated process, and even students with great grades and MCAT scores get left out. That's why more students than ever are turning to Med School Coach for admissions advising. Our advisors are all physicians and former admissions committee members, so they know exactly what medical schools are looking for. One-on-one admissions advising from Med School Coach makes all the difference. Our expert team will help you develop a game plan, prepare your application, edit your essays, and coach you for interviews. Every pre-med has a story, and we'll help you tell it so you can stand out from the crowd. Visit us online at medschoolcoach.com and use offer code PODCAST10 to save 10% up to $400 on a Med School Coach admissions advising package. You can achieve your medical school dreams, and Med School Coach can help.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.